So what really intrigues me about RNA interference is that it has this really deceptively simple molecular basis, but it impacts this huge variety of biological processes, including gene regulation, antiviral immunity, and genome integrity. So, like most molecular systems, we view RNAi through a very narrow lens defined by a relatively small number of very well-studied model systems. And so when we view a molecular system through this type of lens, the function in these different organisms appears very idiosyncratic. And so what we try to do in the lab is use evolutionary analyses to understand the similarities and differences in molecular function across not only these model systems, but all of organisms. So what do we know about how RNAi works? So, like I said, RNA interference has a deceptively simple molecular basis. It starts with a double-stranded RNA molecule. And this molecule is detected by a dicer enzyme that then cuts it at a specific length and then releases it. Now, one of these strands gets attached to an Argonaute protein, which basically creates an effector molecule. And what this effector does is it uses this RNA to identify sequence complementary RNAs and then either degrades them or otherwise regulates their functioning. And conceptually, that's it. That's what RNAi does. Of course, there are complications. One of the complications, the one that I'll be talking about today, is these double-stranded RNA binding proteins, or DRBs. So these DRBs are found really wherever we see RNAi or RNA interference happening. So plant genomes typically encode at least 5 of these DRB proteins. Mammals and flies each encode 2, and the nematode has at least 1 functioning DRB. Now, we know that these DRBs are really essential for efficient RNA interference in vivo, but we have a very poor understanding of precisely how DRBs act to facilitate RNAi. So these DRB proteins are typically composed of a tandem array of 2 to 3 DSRMs. So these are double-stranded RNA binding motifs. And those are the only 2 acronyms that I'm going to use, DRBs and DSRMs. So remember those 2. So these DSRMs can perform 2 different molecular functions, at least. So either, as their name implies, they can bind double-stranded RNA, or they can act to facilitate protein-protein interactions. For example, we know that DSRMs directly interact with dicer proteins in animals and in plants. So there's your functional diversity, and we want to understand how the functional diversity of DSRMs and the DRB proteins that they're in evolve. So these DSRMs are pretty small, and they're found all over the place. They're found in all kingdoms of life, and they're found in a lot of different genes. So the first thing we did was we went to the big sequence databases, and we identified all of the DSRMs that we could find. We then used sequence clustering and phylogenetic techniques to identify those DSRMs that cluster very closely with DSRMs from known DRBs, so things that are from these model organisms. And then just to be safe, we kind of took a slice around these so that we could make sure we didn't miss anything and we could have some nice outgroups for phylogenetic analysis. So what we wanted to do is we wanted to build a DSRM domain tree. So like I said, these DSRMs are very small, and there are a heck of a lot of them. So it makes them a little bit tricky to deal with phylogenetically. The nice thing is that there's a lot of structures for these. So what we could do is we could take all of the structures and do a structure-based alignment, build some maximum likelihood phylogenies, and we used a bunch of techniques to assess the robustness of these phylogenetic estimates, but I'm not going to talk about that part. So we did this, and this is a maximum likelihood consensus tree of the DSRM domains. And so what did we find? The first thing that we found is that the DSRMs from these DRBs all grouped together. So they excluded all the other DSRMs, except for animal Stauffen proteins. So these Stauffens seem to be very closely related to the DRB proteins in animals. So the second thing we found was that the DRBs from plants and the DRBs from animals were respectively monophyletic. So there's a plant clade and there's an animal clade. Within those, the individual DSRMs were also monophyletic. So like I said, the DRBs are a tandem array of DSRMs, and so we can number them 1, 2, 3. And we found in animals and in plants, the DSRMs clustered together. So we have a nice clade of animal DSRM3s, DSRM1s, and DSRM2s are a little more ambiguous. And the same thing in plants. We see this well-supported clade of DSRM2 domains and a little bit more ambiguous of a clade of DSRM1s. And so if we take the gene context and map it onto this phylogeny, we can identify which DRBs these DSRMs came from. And so when we do that, the DRBs segregate into these DSRM clades. So within animal DSRM3, we see representatives of each of the DRB types. Same in DSRM1 and DSRM2 clades, and the same thing we see in plants. So within DSRM2s, we see DRB1, 2, 3, 4, and 5 all in there. So what does this actually tell us about the evolution of this protein family? First thing we found is that the DRBs are monophyletic, they all group together. The second thing that we found was this tandem DSRM architecture seems to have evolved very early and independently in animals and plants. And so we have this situation where there's an ancestral DSRM domain. After the animals and plants split, we get the independent origin of the DRB protein, this domain architecture. And then later on, we see gene duplications of entire DRBs to grow these protein families. But they're independent protein families in animals and plants. So that tells us how the protein family evolved, but we're really interested in understanding how the function evolved across this tree. So to understand the evolution of protein function, typically what we would do is ancestral sequence reconstruction. So we'd use the phylogenetic inference to reconstruct what the most likely ancestral sequences were, and we could then bring those into the lab and study them just like any other sequence. That works really well if you have a small number of sequences, but we wanted to do this across the entire tree. So we use structural modeling and molecular dynamics to infer what the likely ancestral structures look like and to watch them as they interacted with RNA molecules. And we use the structural information and statistical machine learning to then predict the RNA binding affinity of ancestral and extant DSRMs. And so we could then lay this information onto the phylogeny. So this is the same phylogeny as before. We've colored the branches so that red indicates high affinity for RNA. So a red branch is a protein that binds RNA very tightly, and a blue branch is a protein that doesn't bind RNA tightly. So it probably functions in protein-protein interactions. And there's a few things that we see from this tree. First of all, we do see some clades where function is conserved. So for example, this DSRM3 clade, we see that it evolved very tight RNA binding and then maintained that function throughout the clade. And similarly, in this DSRM2 domain of DRB1. But in addition to that, we see a lot of instances in which the function seems to be changing much more rapidly and sort of randomly across the tree. So we see a lot of red interspersed with blue, indicating that the RNA binding function of these proteins can flip-flop between tightly binding RNA and not so tightly binding RNA as they evolve. And so we can further sort of quantify that. We can test for breakpoints on this tree and say where in this tree is there evidence that the function is actually shifting. And so after controlling for multiple tests, we plot these little arrows on every branch on the phylogeny for which there's strong support that the RNA affinity has changed on that branch. And again, so what we see is that these are pretty much randomly distributed across the tree. They're not all clustered here in the middle, they're not all clustered here in the tips, and they're not clustered within an individual plate. They're all over the place. Again, suggesting that the RNA affinity of these protein domains can flip-flop as they evolve. And so, our understanding, at least of the mechanistic basis for molecular functional evolution, has really focused a lot on gene duplications. And we do that, I think, because we expect that gene duplications are going to give rise to functional diversity. But taking a sort of systematic approach like this, we can see that perhaps molecular functions can also evolve uncorrelated to gene duplications and may flip-flop between different functional states relatively quickly. So of course, these are all predictions at this point, and we need to confirm at least some of these affinities in vitro, so we're doing that. Second, this looks at sort of one side of DSRM function, its interactions with RNA. Now we know that these also interact with specific proteins, and so we're doing similar studies to understand the evolution of protein-protein interactions. And then finally, one of the things that we're doing is we're taking these ancestral proteins and we're putting them into extant organisms to try to understand the phenotypic consequences of molecular functional evolution. And so most of this work has been done by a grad student of mine, Raquel Diaz, who implemented and developed these statistical machine learning approaches, and an undergraduate in my lab, Austin Manny, who did a lot of the structural modeling work. And we received some funding for this project from NSF-MCV. Thank you very much. I'm happy to take questions, and I will leave this slide up in case anybody is interested to learn more about what we're doing. Thanks. And no, it didn't come out that well when I practiced. Any questions? Or your bad jokes would be fun. All right, thanks a lot.